Daily quizzes. We know we have daily quizzes. Today's our first daily quiz. We can't make up any other daily quizzes after last week unless you were absent with an excuse. But we had a daily quiz today. If you messed it up, if you got less than 100%, you can come and make it up on Monday or remediate it on Thursday morning for this first period. So that's daily quiz. Tomorrow you have an exam. Exam tomorrow. No remediation for it. These are 100-point quizzes. Tomorrow's exam is 1,000 points. Real simple. It's 20 questions, 20 to 40 questions long, depending on how complicated the questions are. It's all period long. So my question is, what's the exam about? Mitosis, meiosis in general is a good answer. So let's talk about that. Let's say mitosis and meiosis and the cell cycle. Those are the three. Someone want to close it? The door? You can do whatever you like. You get the pass, and if you miss anything, it's on you. Okay. So mitosis, meiosis, cell cycle. What's going to be on the test specifically? Yeah. The checkpoints. So, and let's just put on all three. So cell cycle checkpoints. Where do they happen? What happens? So it's not just the checkpoints. So when we're looking at the cell cycle checkpoints, we got to think of, what are they? You got to think of how do they work? You have to ask yourself what happens when they don't work. So there's some pretty key concepts there. So it's not just as simple as mitosis. It's a checkpoint here with the cell cycle. And how do they work? What happens when they don't work? And what are they? So that's the checkpoints, okay. What else? So the phases, right? In mitosis, you have prophase, metaphase, anaphase, and telophase. There is prometaphase, it does exist. We could describe the differences between prometaphase in most cases, you can think of prometaphase as being part of prophase. It's in between prophase and metaphase. You can put it, these are arbitrary lines, like the states, Ohio, Kentucky. They're just drawn by humans. There's no actual division. They might actually have the line painted, but there's no actual division between Ohio and Kentucky. And it's the same with these phases. They're arbitrary. We decided them based on our studies of mitosis. And what about meiosis? What are the phases of meiosis? Prophase, metaphase, anaphase, telophase. 1. And then prophase, metaphase, anaphase, telophase. 2. Together, don't forget there's a third, there's really something else you can add because it's part of the cell cycle, right? So we can talk about the cell cycle being part of, there's interphase, which comes before mitosis or meiosis. And then there's cytokinesis. And cytokinesis in these phases are known as M phase. So, broadly, with a broad stroke, if we're painting with a big brush, we can see that we have mitosis and meiosis as being part, and all the phases and all the different changes, right? What is it you should know about the phases of mitosis and meiosis? Let's talk a little bit about that. You should know the cytokinesis is a dividing of the cell. You should know what they look like. You have practice questions on all those. I printed them and handed them out to you. You had a POGIL that went through and explained everything step by step as well. You should know how the M phase fits into the cell cycle. How does interphase fit into the cell cycle? You should know the difference between prokaryote cell division versus eukaryote cell division. I hate when yeah.
Should we know how binary fission works?
Not specifically, but just know that there's something called binary fission. And that prokaryotes are simpler. They have less to deal with when they're dividing, so they don't have as many steps, where eukaryotes use the cell cycle. The cell cycle is required for cell division. And there's two reasons to divide. There's only one reason for prokaryotes to divide. What is that? Reproduction. That's all they do. That's the only reason cells divide if they're prokaryotes, to make more of their species, to make more of their larger number of their population. Eukaryotes can use single-celled organ. There's two types of eukaryotes. There's single-celled and prokaryotes can use single. First of all, prokaryotes all of them do mitosis. Right? Single-celled organisms use mitosis for reproduction. So, and eukaryotes, remember, what are we talking about in this chart that I'm drawing here? We're talking about eukaryotes on this side and prokaryotes on this side. So when it comes to eukaryotes, single-celled versions of eukaryotes, what are some of those examples? Yeast, right? Protists. So those are two prime examples of single-celled organisms. They use mitosis for reproduction. Are we good? All right. Meiosis. Meiosis is done in multicellular. Meiosis, I should divide this. I really should have divided it differently, but multicellular organisms. What do I mean by multicellular? And more than one cell. And what does that look like? What are creatures that have more than one cell, what do they look like? Like how are they organized? Are all the cells the same in a multicellular organism? No, they differentiate, don't they? They differentiate into tissues and organs, usually, some kind. They can be very simple. They don't have to have organs, but they're definitely going to have different tissues. So they do meiosis. Multicellular organisms do cell division, and they do meiosis. The multicellular organisms do mitosis. Multicellular organisms, when they do mitosis, their mitosis, if they use it to grow, they're going to use it to grow. They're going to differentiate. You know what? Let me rephrase that. They can grow. They can use it to develop, right? As they become a full adult from one single cell to multicellular. They can use it for repair. And of course, if you have more than one cell, you might have to replace, right? So replace. So you have to consider you're replacing, repairing, and growing. If you're developing and growing and developing, then these two words are kind of associated with one another. If you're growing and developing, then you really need to think about the idea that you're differentiating. Right? They're becoming different. So you're going to develop tissues and organs. The DNA is the same, though. The DNA stays the same. Yeah.
How can one differentiate through mitosis if it only produces identical cells?
That's right. The mitosis, so that's a really great question. So you get a cell that divides into two cells, right? And you're right, they're identical. The DNA is identical. What happens after it's divided? So let's take a look quickly. We're going to zoom in. You know what? Let me make sure you understand. Let's zoom into the nucleus. And in this nucleus, this is the nucleus here. You have this DNA molecule. There's 23 copies, right? I'm just going to look at one. In this cell, this gene is on. This gene is off. That's right. It's epigenetics is what changes the cell. So when we look at what genes are being expressed and which genes are not in this cell, this is what's happening. Now, when we look at the other cell, they were identical. They still are identical. They start genetically, they're identical. Again, it's great we have a pair of twins in this class because they're a really great example. You will be different. You are different. You will continue to differentiate. Not because of your DNA. Your DNA sequences will be exactly like each other. God help you if one commits a crime and the other one gets, there's these DNA evidence, the other one gets busted for it. You better have an alibi at all times. Kidding. I was actually watching a show where they did that. It was triplets. Is it correct? Huh? There were triplets. There's somebody committed a crime, a murder actually, and the triplets were covering for each other, and it was funny. It was just a funny thing. It's a new show. It was interesting, but I could tell you later if you like. It was really funny. So I know murder's not funny, but in this case it was. All right, so you have exactly the same DNA here, but here this one's on and this one's off. So, and now I'm making this very simple. You have to consider that you seriously have a 6-course breakfast with like sauce on your sandwich and everything, huh? And your juice. It's very interesting. So anyways, this cell becomes, brain, a neuron, and maybe this cell becomes, liver, right? 2 different organs because you have 2 different sets of genes. But I'm making it very simple, but you have to understand there are 10,000 genes. And it's not just which ones are on and off, but how on they are and how off they are. You have some genes that are never turned on, like the gene that allows for hair to grow all over our body. We don't do that. But there are other genes that come on, produce some protein at a certain point, and then turn off, and then turn back on later, repeatedly on and off throughout your lifetime. How well that works and how much of that protein is present influences how much of the gene product, how much of the enzyme product that is present, which then influences other genes, or influences the production of tubulin, which influences the shape of the cell, which influences what enzymes it makes, which influences its function. So it's can really get, like, you could go into it and we've worked it out where you can see these really complicated maps of all the epigenetics on a specific cell and all the changes that happen. There's all kinds of names for each protein and enzyme, and there's so many that we have to number them. Like astronomers have to number the stars because there's too many to give names to. Well, it's the same with the proteins in your body. So it's a complicated concept. It's a complicated actual procedure to make it work, to make this creature that's sitting in front of me do all the amazing things it does, all the thoughts and emotions and actions you do. It takes a lot of cooperation between all these different cells. So it's not just turning them on and off, it's also getting influenced by the cells next to them. It's also influencing other cells. So that combination of environment, genetics, epigenetics, all together can make the cells change their function. But genetically, they're identical. Every cell in your body genetically is identical. So mitosis does produce identical cells, but they can change. They can differentiate. This differentiation can be permanent, like in neurons. It's very hard to make a gene turn all its genes back on again, or have the ability to turn their genes back on. We figured it out. That's what we call cloning, right? We could take a skin cell from you and shock it and put it under certain conditions and make it turn into another you. It's possible. But it's a lot of effort, and and certain cells can only go back so far. Some cells you can't take them all the way back to their original state. That's why it's good if you have babies that you save their umbilical blood, because their umbilical blood have what are known as omnipotent cells. So if you, ladies and gentlemen, have babies, I know you can talk to Miss Dalver, and she had triplets, and she had saved their blood, because the blood, when you're a baby, has the maximum amount of potential to become any tissue, because it's undifferentiated. And the umbilical blood has those cells that have not turned any of them on or off yet.
So it's like universal.
It's like a universal cell. And that's why early on scientists used fetuses, aborted fetuses. Why did they use it? Why was that even a topic? It's like, well, they're aborted anyways. And by the way, miscarriage would probably be a decent tissue to get to study. But remember, if they miscarried, there's probably a cause that may not be environmental, may be genetic, right? So aborted fetuses were a good source, although controversial, and the ethics of it is for you to all debate and think about. But it was used to really study this idea of how do we develop, because those aborted fetuses had purely high potential to become any tissue, those cells in that fetus. Does that make sense? All right. And remember, some of the fetuses were aborted because of necessity to save the woman's life. Remember, some abortions are required in order to have both parent and child survive, or one of the two to survive rather than both. Actually, no, just the parent, because the fetus is gone. Yeah, but you realize the mother's dead, the fetus dies as well, correct?
Yeah.
Okay, so as long as it's clear. Yeah.
Okay, so when you said like the DNA has like a gene that turns it on and off. So would like for people like that have freckles, would if like they have faint freckles, would their gene be like turned on but low?
That's a really good, it's a good question. I'm going to defer that to our next unit after tomorrow's exam, but I'll give you a short answer. And the short answer is that having freckles at all is probably genetic, like having the gene for it. But how you express them might have to do with environmental influences. So if you go into the sun a lot, you'll express more freckles. As you age, the number of freckles can change on your face. They can become more, they become less. That could be influenced by epigenetics, which genes you've turned on or off. That could actually be influenced by whether you smoke or not. It could be, that's how smoking could increase your wrinkling of your skin. Like, how does that happen? You're changing the environment in which your cells are existing, and genes are turned on and off. In fact, we know that it ages you. We call it cellular aging. So a lot of discussions here. There's some of you are on your phones. I'm recording this, so I want to have this evidence. A number of you are not writing anything down at all, and we have a lot written down here. All right. Some of you are having 6-course breakfast. I get it, but there's a lot of you that are not writing anything down. Yeah. This is your time. Look, there's no reason to comment. This is your time. I'm just stating the fact for the record, literally for the record, the recorded event. So when the bad news happens, because it's going to happen to somebody out of 134 kids, it's going to happen. That parent, conversation with that parent is going to be, look, let me send you this clip of this audio of me telling them the day before the test, going through and reviewing for the test. And this is them not putting any work into actually preparing for the exam. So their outcome was predestined. They chose it. It's okay with me, as I'm saying here for the record, it's okay that you choose it, but it's your choice. I'm not fighting you. I'll let your parents fight you. Yeah.
Have you ever done any research on gingers?
And people love to use that word gingers. You mean redheads, yes?
Does it seem like they're like kind of going extinct in a way?
No, that's not true. It's a recessive trait, and so going extinct is, first of all, you can't go extinct. They're not a species.
Here's Encanto by James E. Jackson, INCMEA 2010, Northern Region is on it.
It's really freaky that Siri listens all the time. I'm just saying. Like, why? Why would it start playing music? I have no idea why it does that. So, that's not true. Individual parts of the population don't go extinct. Like a species can go extinct, which means there's none of us left. Is that clear? Now, can you get less redheads in a population? Absolutely. Any recessive gene can become less over time if the dominant gene is present more often, right? I have blue eyes is recessive. If I marry someone who has dominant genes, 2 copies of the dominant gene, not brown eyes. I can marry somebody with brown eyes and have blue-eyed children. That's possible if they have the recessive allele. So again, that's next unit. We'll talk more about that later. Okay, that's not on a test, so let's stick on what's on a test. But it's a good question.
But is there actual evidence to show that?
It does not. It is unlikely. No, I would argue that I don't know. Maybe you read something I didn't. It's possible though. For instance, one thing we know is that the Y chromosome in our species is getting smaller, and scientists think that at some point it might disappear altogether, or some other evolutionary change is going to happen. But we know the Y chromosome is getting smaller as we look at older DNA and DNA of our DNA now, our Y chromosome has actually gotten substantially smaller. Yeah.
I had a question about the copies of meiosis. So, because originally meiosis one, there's 23 chromosomes, and then there's 46 copies. So does that mean that that's it just confuses me.
Right, right. No, I'm glad you asked. See, that's a great question because that's really on task. All these questions are great, but the on task ones are most useful. Let's just use one chromosome as an example. All right, so let's just use one. And remember that this is going to happen 23 times or 46 times, right? Let's use two because it's easier to see if with two, right? So you have two chromosomes. I'm going to make one. I'm going to start with one here. This one came from, I'm going to color code them if you don't mind. And it's going to be sexist, I apologize. But the red one's going to be moms, and the blue one's dads. All right? Fair enough. And this is A. And dad is heterozygote. This person's heterozygote. Mom gave him a little a. His mom gave him a big A. His dad gave him a little a. They're heterozygote. Big A, little a. This is a foreshadowing next unit. Dad, let's look at a little smaller chromosome. Chromosome 2. It's smaller. The chromosomes are different sizes. Y being the smallest of the chromosomes, or one of the smallest chromosomes. It's very unique. Y has very few genes. The only thing it really is doing is, which makes sense if you think about it. If you had anything really important on that Y chromosome, why would that not work? If there was a gene that was on the Y chromosome that's very, very important, that you was required to live, why don't you three separate? Why don't you get off your phones before I take them away and separate? I've given you fair warning. Take your phone, put away off your phone, separate now, and start taking some notes. At least pay attention.
I got all the notes.
You're not participating in the conversation. You're giggling and talking to her. That's what when I lift up my head and I look at you, I see you looking at her, not me. Go ahead. What's your answer?
Because there'd be a substantial evolutionary advantage, between men and women, or
That's really, really great idea. You're absolutely right in a very abstract way. But here, if there was a gene that was absolutely required for survival, that was only found on the Y chromosome, why is that not possible?
Because wouldn't everyone have to have
Everyone would have to have it. And since men only have the Y chromosome, the only genes you can have on the Y chromosomes are genes that you can do without to live. I mean, that's the only thing that the Y chromosome can be used, really, realistically. I mean, there are some other things, by the way. It's not that simple. But if you think about it logically, the only real thing you can use the Y chromosome for is to determine sex, male or female. What about the female? I keep telling you, you can't have more than one copy. If you have too many copies of genes, you produce too much protein. The people get sick. Like Down syndrome is trisomy 21, having 3 copies of the chromosome. But men only have one. And by the way, you can't have too few, right? If you have too few, then you could be ill as well, right? So let me ask you something. What about the X chromosome? How many do you need to survive? One. You only need one. Well, wait. How about women? They got one whole extra.
that's because the Y chromosome is arbitrary for survival.
Right. The Y is arbitrary. So X is absolutely necessary. One X is necessary. Without an X, you're dead. There's a very important genes on the X. So, but women have 2 X's. So why doesn't it cause them problems? Because 1 X is all you need. What happens when you get 2 X's?
Maybe because duplication of that necessary genes in the X chromosome is not adaptive. It's fit for survival.
Yeah, you're right. It would be fit for survival. It is. But duplicating it doesn't work because you have too many copies. Here's what women do. This is what our species has done, and most species do this. One of the X chromosomes is arbitrarily shut off. It's completely turned off. The whole chromosome's off. It gets wound up and it's never unwound again. Arbitrarily throughout your whole body, a female's body, every cell just as after mitosis, one of the 2 X's gets shut off. like throwing a net over someone and tighten it up. You ever see those sci-fi movies where they throw those nets over them and just like it tangles them up? That's what happens with one of the X chromosomes.
What happens if both of them are on?
There you die. Their cell dies. There's chromosomes on that gene, on that X chromosome you need for survival. Yeah, genes. I'm sorry, on that X chromosome. Yeah.
Is there anything like that with the Y chromosome in that?
No, the Y chromosome. You can be born with X and no Y, and you would just be phenotypically female at birth. You wouldn't develop into. Well, maybe you would. I'm not sure. I'd have to look at that, but. You wouldn't. You likely wouldn't be able to have children, but you would. What's going on? So you would be phenotypically female, but other than that, you would survive, and there wouldn't be any issues with Down syndrome where you have too many chromosomes. Does that make sense?
Yeah.
And I think that makes sense logically if you think about it. All right. So you asked about meiosis. So let's say that here you have a big B, and then here you have a little b, and I'm just arbitrarily choosing letters. We always use A, B. They're just in sequence. I can use Y, and I don't want to use Y because of the Y chromosome. I don't want anybody to get upset. I can use big Q and little q, and big R and little r, right? They're just variables. It's random. So you have two chromosomes, different genes. Who knows what they do, whatever they do. Could be hundreds of genes on a chromosome. Could be 1,000 genes on a chromosome. Could be 5,000 genes on a chromosome. I don't know. All right? So what is A and B? A might be this really long sequence of A, T, G, C, A, T, 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 A, G. Just a sort of long sequence of DNA, all right, that starts and stops and produces a protein. We'll talk about that next unit as well. So anyways, we got these two sets of chromosomes, 2 copies of each chromosome. That's a normal cell, correct? But we want to do meiosis. So we take it through the cell cycle. The cell becomes bigger. What else happens? In G1, it becomes bigger. What happens in S phase? It's duplicated. So there's going to be 2 of these, correct? There's going to be 2 of these, correct?
Yep.
There'll be 2 of those. How many of these? 2 of these, 2 of these, 2 of these, and how many of these? 2 at the end. 2 of everything. We're duplicating the DNA. All right. So we go through G2 and it grows. Again, checks, everything's checked. Go through 2 checkpoints. Checkpoint between G1 and S. Checkpoint at the end of G2. Those 2 checkpoints. There's going to be another checkpoint in the middle of M phase. The metaphase is checking to see if the chromosomes are attached appropriately. If they're attached inappropriately, stop, freeze, hold the horses. Stop the presses. Yeah.
Do we need to know about the process of interphase between meiosis 1 and meiosis 2?
No. Just know it gets there's something that happens. It checks. You check. You grow a little bit, and then you go right in. The key to meiosis 1 and 2 is you really not need to grow that much, because you don't. Only one of the cells in an egg. Only one of the cells is going to turn into a full-fledged working egg. The other 3 will shrink. They're going to donate their cellular material to that larger cell. The sperm never develops into a full-on useful cell. It develops into these tiny little DNA rockets with a lot of mitochondria and motor proteins. That the only job is to find an egg. Go. And if they don't find an egg, they die. They're cheap to make, and we produce millions of them. So. And that's why men will create them till they die. It's so easy to do, it's not a problem. Okay? But eggs are more complicated because they require so much. There's so much. Your mother gave you so much more, technically, than your dad did. The DNA is obviously even, right? All right, so when you're dividing, now we're in the prophase. Now we're going to get into M phase. You go into prophase. Now the DNA looks different, right? So remember, we went through G1, we went through S phase, we went through G2. There was 2 checkpoints. We had checkpoint 1. And checkpoint 2. That's my alarm letting me know that class is almost over. So much for giving you time to study. We're going to end in 2 minutes. So this is a good question to end on anyway. So you're going to end up with 2 copies, I said, of this one. That's what it looks like in mitosis, right? 2 copies. And there's a B. And then there's going to be, these are called sister chromatids. And there's going to be 2 copies of the father's chromosome as well. But remember, they're going to form a tetrad, correct? And the same with the second chromosome, chromosome number 2. You have 2 copies now, held together by a centromere. It's smaller as well. And am I drawing it so that these 2 chromosomes are overlapping? What do I call that? Crossing over. That's key. And by the way, I don't want you to think that Dad will always give a small r, a big R. The big and small versions of the genes are random. So the crossing over is going to exchange DNA. So you asked the number of chromosomes. Let's be clear. There are still only 2 chromosomes, 2 big chromosomes and 2 small chromosomes. But there's 4 copies. 1, 2, 3, 4. And in the end, 1 copy randomly will be put into a sperm or an egg. That independent assortment. All right, tomorrow's quiz will be a challenge. I hope you study. I hope you paid attention. Good luck. Now, in this exchange of DNA, what you notice, and they start to separate in anaphase and metaphase, they line up as a pair. Because they line up next to each other instead of a straight row like in mitosis, the pair called homologs have the same genes on different, the homologs get separated. So now look, instead of big A, big B, big A, big B, you have big A, big B, big A, little b. Now here, instead of, you still have little b, a little a, here though, you have big B. Here you have little a, and here you have little b. Over here, you have big Q. Here you have big R. Here you still have big Q, but here now you have little r. Here you had little q. Here you have little q. And here you have little, wait a minute, let me double check this. Yeah, this should be big R now, and this one's little r. So you've mixed up the DNA. Do you see that? Now let me ask you questions. How many chromosome number 1s are there in this cell? How many chromosome number 1s are there in this cell?
8.
No. So let me number them. So this is chromosome number number 1 has big A on it. Chromosome number 2 has Q's. So chromosome number 1 has A and B. Chromosome number 2 has Q and R. 2 chromosomes, that's right. So here you have 2 chromosomes. You have 2 chromosome number 1s. Everybody see that? You only have 2. 1, 2. How many copies of the DNA for chromosome number 1 do you have? How many copies of the DNA for chromosome number 1 do you have? 4 is correct. Here's 1. There's 1 right there. There's another one here. Here's another one here. So the sister chromatids are copies. There's only 2 chromosomes, but there's four copies of the DNA. This is the quiz that you were just asked this morning. Here's this chromosome number 2. How many different chromosome number 2 do you have? 2 is correct. How many copies of the DNA do you have for chromosome number 2? 4. 4 sister chromatids. 1, 2, 3, 4. Now, let's be clear. If this creature normally has 4 chromosomes, that's how many it has. How many chromosomes does it have right now? It normally has 4 chromosomes. How many chromosomes does it have right now? Say it again. 4. Still it has 4. How many copies of the DNA does it have? 8. So that's the trick. You have to know the trick. Now when you look at when you separate them, what you end up getting is after cytokinesis is a big A, a big B, this is a big A, this is a little b, this is a big Q, this is a big Q, this is big R, this is little r, this is little a, this is big B, little b, this is little q, this is big R and little r. Now let me ask you something. In these daughter cells, these are called daughter cells. How many chromosomes do you have in each one? Say again? 2. There's 2 in each one. How many do they normally have? How many do they normally have? We just said it. 4. Usually have 2 copies of each chromosome. So now you have, so what do we call these cells? Are they diploid or are they haploid? Diploid means 2. Haploid means half. They're haploid. They have half the DNA. They normally have 4, and now they have 2. Why is anyone putting anything away? My alarm has not gone off. My alarm has not gone off. We're still working. You being distracting is not good for you. So now you're going to go ahead and do meiosis 2. You're going to line them up like you did. Please don't put anything away. Your test is tomorrow. I don't want to hear that you didn't understand if you're not. Wow, just going to go ahead and just giggle away, huh? So now here you had 2 chromosomes each, but you had 4 copies. Here you separate the sister chromatids. So now each of these cells has only 2. They're haploid. All of these cells are haploid. Ploid means chromosome. Half means half. We still got 2 minutes. My alarm just went off. So now we have a big A and a big B. Here we have a big Q and a big R. Here you have a big A and a little b. Here you have a big Q and a little r. Here you have a little a and a big B. Here you have little q and big R. Here you have a big A and a little b. And here you have a little q and a little r. Here you only have 1. They're still haploid. 1 chromosome each of the 2 chromosomes. So 2 chromosomes total. But also you only have 1 copy. You separated the sister chromatids. They're mixed up. Each one of these is different. Look at them. None of these are the same. This one is big A, big B, big Q, big R. This is big A, little b, big Q, big little r. This is little a, big B, little q, big R. This is big A, little b, little q, little r. Each one of these is different. Now they can develop, that's the whole point of meiosis, create half the DNA and mix it up. All right, have a great day.